Populus balsamifera, and it will be given by Karl Fetter, and also his colleagues Vikram Chhatre and Stephen Keller from the University of Vermont.
Thank you. Before I begin, I just want to thank the New England Botanical Club for organizing this and making this a free and accessible venue for graduate students and other people to present. I'm a first-year graduate student in Stephen Keller's lab, and I'm from North Carolina. I just moved up here in August, so I'm glad to meet the New England network of botanists. So I'm interested in the evolution of local adaptation in plants and how do adaptations evolve, and trying to find the loci and the genes that are responsible for local adaptation. Local adaptation is inferred when you have higher fitness in a population's home range versus another part of its range. So here in the north, mean fitness is higher in the north, and in the south it's higher in the south. Or you can have relatively higher fitness in the north than the southern population, but sometimes if you establish a transplant in the southern population, it will have higher fitness, but relatively the fitness is higher in the north. So local adaptation can occur by means of natural selection. And for natural selection to happen, populations need to have different genotypes, traits need to be heritable, and genotypes need to vary in their reproductive success. When the environment exerts a selective force, it interacts with an organism's phenotype. And through selection on the phenotype, it selects for an adaptive genotype here that's signified in red right here. And this adaptive genotype is just one genotype amongst this standing pool of genetic variation that is influenced by past selective events, neutral drift, demographic history of the population, hybridization, and probably many other genetic and demographic factors. But when you have selection for a genotype, there can be a selective sweep where the population evolves, and this adaptive genotype is now represented in all members of the population. So this would be a hard sweep. Hard sweeps are cool and awesome and make great science and nature papers, but a lot more of the sweeps you have are soft sweeps, where any given locus that's associated with some adaptive trait just contributes just a little bit to that trait. And so soft sweeps are probably, I think, are assumed to be much more common, but are pretty hard to find. So once you have some kind of a sweep where the standing genetic variation evolves and becomes more homogeneous, the genotypes change, and of course genotypes change for phenotypes. So through this kind of circle of environment selecting on a phenotype, changing the standing genetic variation, and genotypes coding for selected phenotypes, you can get local adaptation. Detecting for selection is pretty complicated, but the basic premise of it is actually kind of simple. So like I said in the previous slide, you have some locus, here the white locus, that is an adaptive locus, and this increases the fitness of the individuals that have it in the population. So there's the population evolves, and the other individuals in this population have this region of the chromosome, and through recombination, the amount of this original chromosome in red starts to decrease around it. But loci that are linked to this, that are also inherited at the same time as the adaptive trait, they're also present in your region of this chromosome. So when you go and select loci, here are 4 loci selected. If you have a locus where everybody has the same genotype, but at other loci you have either neutral kind of random collection of loci, or you can have certain populations where they share the same locus, but it's kind of more or less random. You can use this to your advantage if you want to find an adaptive locus. So this would be a hard sweep right here with a background of neutral or demographic noise. So population demography is really something you really need to take into account into finding adaptive loci. We're really interested in looking at allele frequencies to find adaptive loci. And an allele can become a fix in a population just because the founders of that population had this allele. And so in the north or in the south or somewhere, everybody has it, not because it's adaptive, but because that's just what was there in the standing pool of genetic variation. So after climate change, populations usually move from the rear edge into a continuous range. and they keep moving in the northern hemisphere. They keep moving north, so they get to the leading edge of where their physiological range tolerance is. And these different parts of the range are important to study because you can find different traits. So if you're interested in drought stress, the rear edge is a good place to find adapted loci for that trait. If you're interested in cold stress, going to the leading edge would be important. So a lot of these different dynamics and effective differences in effective population size and differences in population structure are all things you need to keep into account when you're looking to find adapted loci. So my research questions are simple for my system. I work with Populus balsamifera, and we wanted to understand the population structure of Populus balsamifera, and we wanted to identify the core and edge populations and find the number of edge populations that existed and where they're located. And then we wanted to find candidates for adaptive evolution, for local adaptation to environment between these core and edge populations, and also across the entire range. So again, my system is Populus balsamifera. It's a really great system for studying local adaptation in trees. They behave really well. You can collect them in the wintertime, cut them off, and put them in soil, and then do what you want with them. They're a cool system. They grow very fast, so I think in about 5 years you can get a pretty tall tree. Here is the range of Populus balsamifera in gray, and the black are our study sites. I've got a 57 population sample, about 508 individuals. And here's a PCA of the climate data that I'm going to use to find adapted loci. So I'm going to end up using PC1, which is, I'm interpreting this loosely as kind of a dry to moist PC, and it also goes from very highly seasonal to less seasonal in Atlantic Canada. And you can see kind of where the biggest breaks in the data are, and they kind of somewhat loosely conform to different groups of populations. So for our genetic data, we used a type of data called genotype sequencing, or a method for collecting that data. So a genome is large and complex. Populus balsamifera has about 500 gigabases of information. So to cut down on that, we use a methylation-sensitive restriction enzyme. And areas of the genome that are methylated are usually not doing work that we're interested in. They're not coding for physiological responses. So if you cut down on the methylation, you can cut down the complexity of your genome and just target your sequencing to non-methylated regions. So our sequence fragments that were digested by this restriction enzyme were sequenced in Illumina HiSeq, and we aligned our contigs to the Populus trichocarpa genome, and we called SNPs. So we got, from our SNP column, we got 683,000 SNP loci, which we eventually filtered for sites that are out of Hardy-Weinberg equilibrium, low-quality sites, sites that have a lot of missing data, and sites with a minor allele frequency of less than 0.001. So that was to try to get rid of heterozygotes that we thought could have been bad reads. So a read from a couple different individuals aligning to the same place, and we thought this isn't a real genotype, so we filtered it. And our filtering reduced our data set to 167,000 loci. So pretty big reduction, but we still got a lot of loci to work with. So my methods go into two different things: population structure, and then the GWAS, the Genome-Wide Association studies. We use fast structure, which is a maximum likelihood implementation of the popular structure program. It's really fast, really easy to use. It takes about 1.5 minutes to run fast structure on our data set of about 100 million pieces of information. We also use DAPC, which is a R package. This is a discriminant analysis of principal components. You just find the number of clusters in your data set using k-means clustering. You perform a PCA on your data set, and then you perform a discriminant analysis on those PC axes. So it's a great way to reduce the complexity of your data set and to have a similar but different picture of population structure than the fast structure. So for our GWAS, the essence of doing GWAS is finding where your environmental variables, where that covariation kind of syncs up with the covariation in your genetic data set. And when you find correlations, you can infer adaptation. But of course, population structure in neutral loci that are fixed in one region can also mimic adapted loci. So our methods account for that by, for base scan, this is a method that uses FST to find it. So you basically, you calculate a null model of FST for all your populations, and then you look for loci that have very, very high FST among some threshold. For LFMM, latent factor mixed models, you have a mixed model that estimates the hidden, which is what the latent comes in as, the hidden effects of population structure. And this is a Bayesian method that is new and pretty exciting. Base scan is a great way for finding local adaptation because it's agnostic to the source of selection. And LFMM takes a predictor as an environmental data. So you can put in your environmental matrix, and then you find loci adapted to environment. We also can use DAPC to find SNPs. So DAPC, of course, you get your loadings from the PC scores, and you can kind of filter those for the loadings that contribute the most to what's going on in your discriminant access space. So it's a non-parameterized method for rotating the data to see what loci contribute the most to the picture that you have. So here's the first picture that we have of fast structure. K equals 3 was the K that was selected, but of course, one level of K is not the gospel. Looking at other levels is important. The biggest, K equals 4 is kind of weird. K equals 3 and K equals 5 are the interesting ones, because in K equals 3, you've got a group down here in the Rockies, and then a large core group, and then a group in New Glasgow, Nova Scotia. And in K equals 5, you have an Atlantic Canada group that pops out, but these groups kind of stay the same. So DAPC is great because we can see the magnitudes of differences. So Nova Scotia is really quite different from this, but when we kind of zoom in and choose K equals 4, we can see we have a core population, Atlantic Canada, Western Rockies, and the Southern Rockies population. So here are our core and edge populations that we can now start working with. Here is a kind of a GWAS from DAPC. This is a Manhattan plot with a lot of loci above a certain threshold that contribute a lot to this. So these are the loci that differentiate discriminant axis 1, so our core from our Western and Southern Rockies populations, and this differentiates the core from the Atlantic Canada populations. So I'm going to move on. All right, base scan. This is the FST loci. This is loci that are just really different for some reason. But the ones that I'm most interested in are the ones we get from latent factor mixed modeling. So these are all loci that are associated with climate. So above this red line right here, we have about 4,000 loci that are associated with climate with a very stringent false discovery rate. So these loci are cool because this is a whole list of hypotheses that we can start looking at and start testing, or just start kind of digging into it and see what are these loci, what's going on. So here's chromosome 13, and this locus right here is cellulose synthase 3. So cellulose is a really important part of cell walls. It's one of the most, I guess it's what cell walls are made of. And cellulose synthase 3 is highly expressed during the development of cell walls in oomycetes. Here's the expression data for some Populus. I'm not sure what species it is, but it's for Populus. And it's down expressed in young expanding leaves, which is kind of interesting. You think it would be more expressed, but it's actually down expressed. So here's an interesting hypothesis to follow. Another one is a protease inhibitor. It's expressed at kind of background rates in the leaves, but then in mature leaves, it's down expressed. So proteases, mature leaves are frequently attacked by all sorts of pathogens and all sorts of things. So you want your proteases to be active when you have mature leaves that can start doing work. And again, in leaves that are attacked by beetles, you have down regulation or down expression of this protease. So we think that this could be adapted to different environments. How, I don't know. This is the hypothesis that we got last week that we need to start working with. So as you can see, there are a lot of loci that are associated. This is kind of cool. Let me go back. We get hits in the 3' untranslated region here. So kind of the next steps is to start looking at, start describing these loci and seeing if there are any non-synonymous ones, if they're synonymous loci, how many are UTRs, how many are introns, and to start to describe some of this adaptive variation and try to understand what is going on with our traits. So the next steps in the study, we've got 2 common gardens of about 1,000 individuals planted from collected across the range. One is in Burlington, Vermont, the other is in Indian Head, Saskatchewan. So we're going to start doing GWAS on phenotypic data and looking at bud size, bud set, bud flush, height growth, photosynthesis, stable isotope data, and start getting a better idea of what's going on phenotypically when you control for environment and when you also control for genotype between environments. So it's a really exciting study that we have going on. We have a lot of loci that are candidates for local adaptation, and soon, if we find really interesting loci that are cool stories, we can possibly do transgenics on them and see if we can recover a similar phenotype in Arabidopsis or whatever plant you want to work with. And yeah, so this, I think I'm done. Thanks. Oh yeah, acknowledgement. Right. Steve Keller. So Steve Keller is my advisor. He's a new faculty member at the University of Vermont. If you don't know him, if you haven't talked to him, you should come up for a visit. He's a really awesome guy. Chhatre is a postdoc in our lab, and I am bugging him all the time with questions. So is helping with a lot of the work that you saw. And we have funding from NSF and from the University of Vermont, and a lot of collaborators at the University of Maryland. And our lab is really awesome. And thanks.
Questions?
Yeah.
I was just wondering with the Atlantic maritime groups, do you think some of those genes might be from introgression from Populus deltoides? Because when you're out in the field, X. jackii is not terribly uncommon. I was just wondering if that could be where some of the genes are picked up.
I think that might be what's going on right here with this Nova Scotia population, this New Glasgow population. The leaves look a little bit different. I didn't collect them, but I'm told that the leaves kind of look like they may be a hybrid with an Asian populace that has been planted there. So we are going to, there's a SNP assay that was developed by someone in Forestry Canada, Sally Aitken, I think, developed it. And we're going to try to use her SNP assay to find out if these are Populus balsamifera or if they're a hybrid, or it could be a deltoides hybrid. So that's something that an undergraduate is going to be doing probably in the fall to figure it out. And having this huge difference in the data set is kind of problematic, right? If you want to just look at adaptive loci and not have to deal with demographic history. So that's an important thing to find out.
Thank you.
Yep.
Do people have a sense of what's going on at the range edges of this species? So when you're looking at its distribution, like, are there field studies or have you visited populations out at the range edges to see if it's sort of pressing up against its ecophysiological limits or ecological limits?
I don't know. I know there was somebody got in touch with Steve, my advisor, and said he found some Populus balsamifera that was kind of knee height in the Arctic. So it's kind of strange to have it so small, right? But I guess a lot of trees are small there. So it could be moving its range north. I'm not really certain what's going on with that northern range margin. Go ahead.
Differences in, let's say you said that you've got 2 sites where you're on Saskatchewan or whatever. Differences in day length, differences in growing season, differences in low temperatures in the winter of the 2 sites and then the like is.
Yeah, those are, that's exactly what we're interested in. So when you control for the same genotypes and you put things in different environments, what different phenotypes do you observe? And when you have everybody control for their environment, but there are different genotypes, what are kind of the natural differences that you observe in the genotypes? So having 2 common gardens at 2 different places is kind of a key part of our study design, to observe the effect of environment on genotypes, common genotypes.
I just wanted to make a comment with Populus balsamifera in southern New England, and like up in UVM area, it's more of a generalist associated with lakes and rivers and things and wetlands. But as you get further further south in New England, it shifts almost exclusively to calcareous wetlands. And I'm not sure what's going on there, but it's just kind of an interesting observation, and so I'm not sure if there's anything genetic going on, or just if the calcium helps it moderate some of the heat stress. But that maybe an interesting thing to look at some of these southern New England populations that are on calcareous substrates. When I think of it as in northern New England as a soil chemistry generalist.
Yeah, that's interesting. I will kind of report that back to Steve and talk about it with him. Thanks.
Thank you.
Okay, thanks.